Hello and welcome to the 21st Annual Mitsch Lecture. I'm Nick Schlatter and I am the current Chemistry Department Chair at Hamline University. I will be the host for this event. We'll provide some background information and introductions for Hamline's president, the Chemistry Department, students, faculty, and finally tonight's speaker. In a bit I will introduce Hamline's president to speak about the Ron Mitsch Fund, which makes today possible. But first, I want to talk about Hamline's Chemistry Department. Our Chemistry Department is an American Chemical Society certified chemistry department, and we strive to prepare students to be ready for careers in chemistry, but also in other fields that find a technical background important. Whether we're talking about getting into doctoral programs, medical fields, law, business, or something completely different, we have many students that have continued on successfully. We provide our students with research experience as well at Hamline that are authentic and often lead to regional and national presentations and occasionally publications. Our faculty run summer research programs in a variety of areas such as organic synthesis, medicinal chemistry, biochemistry, polymer, plasma matter interactions, nanotechnology, forensics, inorganic materials, and many more. Some of our faculty will give you an update on their individual work shortly, but first I want you to hear from our students. Our current Mitsch Scholarship holders are Natasha Back, Luca Fisk, Soren Kemmer, Ash Robinson, and Cindy Van Acker. I think we can applaud them. We will now hear from some of the students a bit about their Hamline experience and their research.
Hello, I'm Ash Robinson, and I'm a senior here at Hamline University pursuing a major in biochemistry. And I work in the Marti S laboratory. In the Marti S laboratory, we're pretty interested in microbes, specifically how microbes can potentially break down some pharmaceutical compounds that enter the environment. My project has worked primarily on metformin, which is a drug that's commonly prescribed in the treatment of type 2 diabetes. It doesn't get completely metabolized by humans, so it actually enters the environment where it has some hazardous effects. And some of the work that I've been able to be a part of has led to the discovery of an enzyme important in the breakdown of this compound. And this work will be published and applied in environmental microbiology. And it may sound cliche, but Hamline University, the faculty here really do notice the students' hard work, and they also work hard in turn to make opportunities and give opportunities to students. Everything that I've gotten to do has been because there's been a faculty member behind me along the way giving me the opportunity to pursue more when I wanted to. And this opportunity has also been due in large part to some financial support I've received and we've received along the way. In my case, like the Ron Mitsch Scholarship. So I'd like to thank Ron Mitsch for his generosity and support of the work that I've been able to do here at Hamline.
Hi, my name is Cindy Van Acker. I am a senior here at Hamline. I'm a biochemistry major. I have recently been working on developing a 3D model of HIV proteins to explore different kinds of drug resistance within the proteins with duranavir and tipranavir. So one of our models showed just the active site of the protein itself. So you saw all the specific molecules that we were looking at in there, and with that, we were allowed to take off the top to see those molecules a bit better. And then within that top piece, because duranavir uses a water-mediated bond, you can take that water molecule out and kind of see how that drug then interacts with that protein that way, which is really, really cool to see. Being a Mitsch Scholarship recipient has helped me a lot. It's allowed me to see different things within the chemistry field, whether that be new reactants and solvents or new equipment to be able to use. And I just am eternally grateful to Dr. Mitsch himself for giving this scholarship and then also donating to the school itself to allow lots of different students to have these opportunities that they maybe wouldn't have been able to get anywhere else. Thank you so much, Dr. Mitsch.
Hi, my name is Luce Fisk, and I am interested in organic synthesis. For the past 2 summers, I've conducted research here with Rita. I looked into a multi-step synthesis to produce the small fluorescent tag MBD-F to be able to attach it to cortisol. After Hamline, I want to go into industry to be able to grow and explore what I want to do into the future. But thank you, Dr. Mitsch, for supporting me and supporting my research.
And now I would like to introduce our current faculty. Our current faculty are Yarashi Gangle, John Manichek, Rita Majurli, Mark Schulten, and myself. I would also like to acknowledge our lab coordinator, Marina Gorina, who has held the internal workings of the department together throughout all the challenges, especially this last year. Some of our faculty will now say a few more words about their own work.
My name is Mark Skolton. I'm a visiting assistant professor here at Hamline. This year I've been engaged in research in addition to teaching a variety of levels of students, from non-majors teaching Chem and Society to advanced students fulfilling their capstone research requirements. I feel grateful to have research opportunities here. I've engaged in a variety of projects revolving around the synthesis of polymers, particularly polyesters and polyacrylates, in addition to depolymerization projects, which tie into sustainability a little bit more. One of my favorite projects here occurred last summer. I worked with Brendan Hine on a depolymerization project where you got to take plastic cups from the Anderson Center, essentially, and depolymerize those and make basic epoxy resin. And so we even got to make glue out of what would ordinarily be a waste product, and that was really satisfying to see.
Hi, my name is Maddie Cobo. I'm a senior at Hamline University, majoring in biochemistry. I did research with Professor Mark Skolton in the summer of 2019. The main goal of our research was to reduce waste in an organic chemistry lab that was synthesizing aspirin. And from that waste from the lab, we synthesized polysalicylic acid. And then from that polymer, we were able to get cyclic monomers. And with those cyclic monomers, we wanted to perform a ring opening polymerization. And next year, I'll be going to the University of Minnesota Medical School, where I hope to do research also.
Hello, my name is Rita Majurli. I am the organic professor of chemistry here at Hamline University, and welcome to my lab. This lab is where students and I work on research to develop new bioactive compounds, to develop new methodology, and to test materials that we want to know if they're what they say they are. One of the projects we are working on is the DPAL project with the University of Notre Dame. With this project, we look at underserved countries and continents for gray market drugs to see if they're past their expiration date, if they've been adulterated, or any kind of compromise that might affect someone's health. My students have done 2 different types of malarials, and currently we're working on the antibiotic ivermectin to find out exactly how these things decay and what we need to see. Other things we do is look at natural products for the plants around us and what they can do for our lives. For example, here's some prickly ash that I have soaking in vodka to extract the natural products out of it. I have students working on looking at the products that we get out of this stuff. This is a local plant. And we're trying to figure out what may, in fact, contribute to our health. Other things we work on include looking at a particular antifungal and algicide piece called casamigamide. This is a small molecule that kills off green algae. It's made by blue-green algae to do as a defense mechanism. And because of that, it is a natural product that we could possibly use to control algal blooms in the state. One of the things my lab is interested in is real-world applications to problem solving and new products and design for things. Students with me get the chance to do undergraduate research from their second year on, and we go and present at national meetings, we do publications, things like that. So we want students to come on in and feel at home and find their vocation here at Hamline.
Hello, I'm Nick Schlatter, and I've been a chemistry faculty member at Hamline University since 2000. I got into academics after working in industry at IBM and Bell Labs. In those days, we were looking at thin molecular films, something we now call nanotechnology. In fact, I'm still fascinated by nanotechnology, and one of my current projects involves using nanotechnology and spectroscopy to detect biomarkers. Biomarkers can be a lot of different things, like cell signaling molecules from the surface of bacteria or the coronavirus. What they have in common is they can be a very small fraction of a biological sample, and the technique I and my students are developing and working on can capture these minute quantities of biomarkers and detect them. I'm also having fun developing 3D printing to translate molecular calculations of structure and energy surfaces into macroscopic models to help us understand what is normally invisible. There's also the potential to make lab equipment that is both practical and educational.
Hi, my name is Urvashi Gangal and I'm a visiting faculty here in the Department of Chemistry. I teach General Chemistry 1 and 2, Physical Chemistry 1 and 2, Instrumental Methods and Chemistry in Society lecture and laboratory courses. Besides teaching, I also do research in plasma chemistry, so I mentor undergrad students. So this is our plasma setup here. This project is in collaboration with Department of Mechanical Engineering, University of Minnesota, and we have a plasma source that has been designed by them, and it has been given to us so that we can carry out some chemical studies with it. Plasma is the fourth state of matter, and it's an ionized gas, so we pass a high potential between electrodes having a gas and a discharge is generated. It's like lightning striking the atmosphere. And we are using it for environmental purposes, like we are treating water with it and trying to inactivate the harmful chemical pollutants in water. And besides that, we are even doing some material studies, like we are trying to synthesize polymers and nanoparticles. And this group just started in 2019, so it's a new group, and it's called as the Hamline Plasma Laboratory. And plasma is a very frontier field of research, and it's really good that undergrad students can also be exposed to this research, which is primarily done by grad students.
At this point, I would like to introduce Hamline's President Miller to say a few words about the 3M Ronald Mitsch Fund. President Miller, the floor is yours.
Welcome to the 21st Annual 3M Ronald A. Mitsch Lecture in Chemistry. I'm President Fanice Miller, and I'm pleased to welcome you here today for this event. In more normal times, we would see you here on the Hamline University campus. This time, it's a virtual get-together. I look forward to the time when we can all be together again for this signature event offered to students, the Hamline family, and the state's scientific community. Hamline is proud to host many noteworthy lectures on its campus in a variety of disciplines. Hamline University's mission statement, values, and mission are put into practice every day. We are driven to create a diverse and collaborative community of learners dedicated to the development of students' knowledge, values, and skills, leading to successful lives of leadership, scholarship, and service. The 3M Ronald A. Mitsch Lecture in Chemistry exemplifies the values Hamline University promotes. We're pleased today to welcome our speaker, Dr. Marti S, Hamline class of 1991, whose postgraduate path has led her to the Oak Ridge Laboratory in Tennessee, researching and working on the most current problem in the world, using scientific processes in the search for COVID-19 therapeutics and how to get them to people faster. As embodied by Dr. Head, service to our community is a Hamline tradition and a critical part of what we teach. Dr. Ronald Mitsch is a graduate of Hamline University, class of 1956. He joined 3M as a research scientist 4 years later, garnering 19 patents for his work there. When he retired in 1998, the 3M Foundation established an endowed fund to recognize and appreciate Ronald Mitsch's outstanding leadership and service to the 3M company. That gift was matched by Ron and his wife, Marilyn. Together with Hamline University, they created the 3M Ronald A. Mitsch Endowed Fund in Chemistry. Their combined generosity supports this lecture, plus several scholarships and collaborative research grants. Just a moment ago, you met some of the students who are the recipients of those scholarships. On behalf of Hamline University, I want to extend my thanks to Ron and Marilyn Mitsch for their generosity. It allows these students opportunities to do exceptional scholarly work at Hamline and to prepare for careers of service and leadership. Thank you. It is now my pleasure to introduce Dr. Nicholas Schlatter, Chair of the Hamline University Chemistry Department.
Let me give you some background. For Hamline students and faculty, the 3M Mitsch Fund has provided significant support for student scholarships and summer research stipends that have been instrumental in our goal to give as many of our students a possible research experience. The Mitsch Fund has also allowed us to maintain our research instrumentation. 3M and Ron Mitsch's generosity has been incredibly fruitful, with many of the supported students going on in science careers. Thank you, 3M and Ron. Dr. Marti S. Head is our speaker today. In 1991, Dr. Head received her BS in chemistry from Hamline University. Go Hamline. At Hamline, she carried out experimental studies of the factor I mediated modification and release of red blood cell bound immune complexes and developed a computational model of the system, doing systems biology before it was a thing. This undergraduate work seemed prophetic, since her PhD thesis in 1995 at Duke University was doing computational work to understand proteins, followed by more computational chemistry as a National Research Council postdoctoral fellow. Then a career at GlaxoSmithKline, GSK, doing and directing computational work on therapeutic technologies, and is currently a senior position at Oak Ridge National Laboratory with a similar focus. In particular, Dr. Head is currently the director of the Joint Institute for Biological Sciences, a collaboration of Oak Ridge National Laboratory and the University of Tennessee. Dr. Head's original focus at Oak Ridge was ATOM, which stands for Accelerating Therapeutic Opportunities for Medicine, a public-private partnership amongst the Department of Energy, the National Cancer Institute, GlaxoSmithKline, and UCSF under the auspices of former Vice President Biden's cancer moonshot. Due to the pandemic, the focus has shifted to work she has been doing in the molecular design to accelerate therapeutics for COVID-19 project through the Department of Energy's National Virtual Biotechnology Laboratory, or NVBL. Dr. Head is the project lead for the NVBL Molecular Therapeutics Project that is leveraging the world-leading capabilities of the Department of Energy National Labs to look at the coronavirus to develop solutions to dealing with the pandemic. This involves both computational and experimental work to design therapeutic antibodies, and there are nearly 100 or more scientists in this project. Dr. Head has authored numerous publications, presentations, and book chapters in computational and medicinal chemistry, and is listed as the inventor on multiple drug discovery patents. Dr. Head has served on numerous editorial boards in her field, has been a board member for a number of high-profile national science organizations such as the National Institutes of Health, and is a science advisor and board chair for the Dance Exchange. The Dance Exchange seems to connect art to science. Maybe we can find out more about this later. I'm excited to hear Dr. Head's talk, which will be followed by a live question and answer session online. So without any more delay, please welcome Dr. Marti S. Head.
Hello, it's a pleasure to be back at Hamline again in this very virtual sense. And it's a real honor to be invited to give the Mitsch lecture this year, especially after having the chance to look back at who's spoken at the Mitsch lectures over the past few years. And as many of you may know, the original plan was that I would be there in person last year to give this presentation. Hasn't quite worked out that way. If we had been together last year, the old title of the talk would have been On Transformation of Drug Discovery, a talk in 3 parts. And those 3 parts would have involved the narrative of my journey from Hamline to where I am today. It would have included a second part that is a brief introduction to the traditional drug discovery process as practiced today. And it would have included a third part that's about a public-private partnership that aims to transform the way we do the early stages of drug discovery, from the first time you have a biological hypothesis through to when you have a small molecule that's ready to go into clinical trials. That public-private partnership involves the DOE national labs, it involves the National Cancer Institute, and it involves University of California, San Francisco, and GlaxoSmithKline Pharmaceuticals under the auspices of Vice President Biden's Cancer Moonshot. But we won't go into any more detail about that today, other than to say, I look forward to talking to you again in the future about that. For today, though, we're shifting gears. Instead of in person, we are here at this virtual Mitsch lecture, and there is a new talk title, Responding to a Global Crisis, a talk in 2 parts. And so the first part will still be the narrative, the story of my journey through time and space to get to where I am now at the Oak Ridge National Laboratory. And then it will have a new part that is around the work that the Department of Energy National Labs have done in attempting to respond to the crisis that we're all living through at the moment. And so the journey. I began at Hamline University back in the late 80s. As I was looking over these slides, I was astonished to recognize that it's been almost exactly 30 years since I graduated from Hamline. At that time, I was working with Dr. Runquist in the chemistry department and working on the project with the University of Minnesota rheumatology department and Dr. Nancy Merrihew, thinking about the processes by which immune complexes are made, bind to red blood cells, are modified, and then released back into circulation. And this is part of processing immune complexes to respond to insults from the outside to the human body. And there was some thought that there are dysfunctions in that part of the process. And so we were trying to understand, what does the process look like for normal, healthy human individuals compared to what it looks like in patients who have systemic lupus erythematosus. Many of people over the years at Hamline University have been involved in at least some way in this research project. I was there doing actual physical experiments in the lab. That was back in the day when it was still possible for me to draw blood from normal controls without worrying about a whole host of issues, and taking that blood into the lab, doing the experiments, and then calculate using some of the early computing resources at Hamline University, in particular the VAX operating system computers that were also used by the bursars and the finance crowd at Hamline University to calculate the rate constants by which this process was occurring. As part of my senior research, I started down a path that Ole had always wanted to go down, which is building a computer model of that formation and modification and release system so that we could ask what if questions. What if the rate is faster here or slower there? What impact does that have on the behavior that we see in the physiological system, or at least our small model for that physiological system? And so because I was working in computing, because I had done some of this work recoding his algorithm, this is the senior research project I worked on. We were doing systems biology back in the late 80s before it had become a thing. And as I look back on it, if I were to do it again, I certainly would do the research a little bit differently. I would calculate all of those integrals just a little bit differently. But I'm pleased and proud that I had the chance to be involved with this cutting edge research at the intersection between thinking about processes in a chemical way, thinking about biology using that chemical thinking, and using computing to understand the ways the two integrate together. From there, I went on to do my graduate studies at Duke University. I was in the chemistry department, but I was doing a joint project with the computer science department, working with the ray casting engine, a special purpose, massively parallel computer that was first created for the purposes of mechanical design. Can I design machine parts by breaking them into their component bits and understanding how they fit together? My research advisor's key insight was that the meshing of gears in a mechanical device is really topologically very similar to small molecules binding into protein structures. And so my graduate work was really around building the tools to be able to use this machine to understand the geometric properties of protein structures. What you see at the bottom is a snapshot from a simulation of myoglobin, an important protein for carrying oxygen around the body. And we had developed techniques to identify cavities within the protein structure inside the protein and really begin to think about how do those cavities percolate in and out to allow oxygen to move from the outside to the completely enclosed cavity in the inside. And a host of other questions really at that intersection of how do you understand the geometry of a protein structure and how that connects to its biological function. After Duke, I went on to do a postdoctoral couple of years at the Center for Advanced Research in Biotechnology. I was with Mike Gilson there. CARB is jointly affiliated with the National Institute for Standards and Technology and with the University of Maryland, and is in Gaithersburg, Maryland. And in Mike's lab, we were looking at statistical thermodynamics. And specifically, we were trying to develop a method that would allow us to calculate from first principles the equilibrium coefficient for non-covalent association of two molecules. And so you can see how that starts to be important as you think about not only what's the geometry of how small molecules interact with proteins, but what is the thermodynamic consequence of that? Is it a favorable or an unfavorable interaction for that molecule to bind to the protein? And so across the span of my educational career and my postdoctoral time, really had the privilege to get a viewpoint on kinetics and mechanism and on geometry and how proteins work, and then translate that into an understanding of the energetics of those interactions so that we can have a whole picture of what's going on when molecules interact with proteins in physiological systems. And with that backing, that training, that set of experiences, I then moved on to GlaxoSmithKline Pharmaceuticals, becoming a member initially of the computational chemistry group in Philadelphia, ultimately having the chance to lead the computational chemistry function in the United States. So leading a team of really amazing scientists whose job was to apply every computational technique appropriate for moving drug discovery projects from that initial hypothesis through to that clinical candidate selection decision, that same process that we're now trying to transform in that public-private partnership I mentioned before. While at GSK, we spent a lot of time thinking about protein ligand docking calculations, coming back to even the that question of the geometry of proteins back when I was at Duke, thinking about how well do we do with the programs that have been built over the years that calculate these protein ligand docking simulations. And so in docking, we start with a structure of a protein, usually solved by X-ray crystallography, so that we know where all of the atoms in the protein sit. And then we're running calculations to generate many candidate poses for how the small molecule sits in the protein, and trying to figure out what is the most likely orientation that's going to match what you see in a crystal structure. What are we able to use this technique to hunt out in a long list of molecules the ones that are most likely to interact with the protein? And also, can you use it to estimate the energy with which a small molecule binds with a protein? So evaluating technologies that people have created that are tapping into some of these computations and techniques and technologies that I had been learning through my educational career. I'll say that the punchline from that large study, looking at 12 docking programs across 35 scoring functions, looking at 7 protein ligand systems that are relevant to the drug discovery process. And our punchline is that you absolutely can use these programs to identify the poses and ways that those small molecules might sit in the protein, but the scoring functions don't always reliably allow you to tell which one of those is the correct answer. We determined that there are circumstances where you can use this kind of technology to select active compounds from a random list. However, you don't know upfront which docking program and which scoring function is going to be exactly right for the protein system that you care about. And finally, because a docking calculation works on a single pose of the molecule bound to the protein and assigns it essentially a single number, that means that you're missing out on the full complexity of the equation that describes that interaction. And so it is of no surprise that docking programs are actually utter failures at being able to rank order the binding affinity of a set of closely related small molecules. And so it was an important study that the large team across GSK were able to pull off because of the data we had in hand, because of the people in the extended team across all of the research sites at GSK. And we went on to do a small piece of work playing a little bit of a game show to ask, can a human expert do better than these computations? And the answer is that a human expert, in combination with these kinds of computational tools, actually can do better than the computations alone. But it has to be the right person with the right level of expertise and experience with this particular technology that's able to do it. And so lots of fun work. It was while I was at GSK that I got involved in this public-private partnership, got connected to the National Lab community through Lawrence Livermore originally, but also meeting the folks down here at Oak Ridge National Labs in Tennessee. And that led me to what is now my present and my future, my role at Oak Ridge National Labs as the director of the Joint Institute for Biological Sciences. And the picture that you see on the screen is of the large Oak Ridge main campus, nestled into the foothills here in East Tennessee, just outside of the Smoky Mountains. So not only is it a place with lots of amazing scientific expertise, it's also a place that's in a really beautiful part of the world. And I'm going to give you a brief introduction to Oak Ridge and how it fits into the world. Oak Ridge is one of 17 Department of Energy national labs scattered throughout the country. They align with different of the mission spaces of the Department of Energy, either a broad science focus, a very particular focus on energy policy and energy science and technology, a focus on nuclear security, and a focus on the environment. Oak Ridge is the largest of the science labs and embeds most of the capabilities of the National Lab System within this single lab. At Oak Ridge is has a long history here in East Tennessee and a long history as a national laboratory. It is one of the first 3 labs that were created as part of the Manhattan Project. They were integral to that response to a global crisis back in our past. They created the first continuously operating nuclear reactor. And this is the place that did the physical chemistry of separating out isotopes and elements so that they were able to separate the plutonium that would then go on for nuclear energy and for making the atomic bombs. In addition to that rather fraught history with that set of topics, in addition, Oak Ridge National Laboratory was early on in the study and understanding of how radiation and radiation events impact biology and impact biological systems. And they continue to be a place where biological research goes on, as well as chemistry and nuclear research. We execute our mission at Oak Ridge through a set of user facilities and departments and directorates. What I'm showing you here is a list of the user facilities that bring thousands of partners to Tennessee each year to do research and development jointly with scientists and researchers at the lab. Everything from building technologies to transportation, carbon fibers, technologies, and manufacturing, a whole host of things. I'm going to focus specifically today on the Oak Ridge Leadership Computing Facility. What I'm showing you here is Summit, our pride and joy at the moment. It takes up the entire basement of a room, and it was, until very recently, the fastest supercomputer in the world. It's now second in the world, but still fastest in the US. And so we have this huge capability in high-end, high-performance computing. We're moving into the next iteration of computing infrastructure with computing ecosystems, computing infrastructure that mimics the ways we think about brains and bodies working and transmitting signals, and taking insights from the quantum world and building quantum computing systems and algorithms. And later this year, we expect to take delivery of Frontier, which will then be the first exascale computer in the world, and will again win us the first place with the fastest supercomputer in the world. The other user facility I'll bring to your attention today is the spallation neutron source. Oak Ridge is home to the world's most intense pulsed neutron source. Great, what does that do for us? Well, we know that you can shoot X-rays at crystals and understand where the heavy atoms, the carbons, the nitrogens, the oxygens are in that protein. But it's very hard to see, if not impossible to see, more detailed information like where the hydrogens are and where they might be moving as part of a chemical reaction or an interaction or an enzyme carrying out the function that it's built for. And so with neutrons, on the other hand, you really can get down to that resolution of seeing where all of the hydrogens are, and you can begin to understand more about the static structures and the dynamic conformational variations of proteins using this very intense pulsed neutron source. Just one flying example of work that I'm aware of that I find really fascinating here at Oak Ridge. They were able to grow an E. coli bacterial cell in a deuterated medium. And so what they've done is swap out the hydrogens for deuteriums. And now the proteins that have those deuteriums on become invisible to the neutron source and the signal there. And instead, what you're seeing is only the things with hydrogens on them. So they grew up the E. coli in this deuterated system. They added back in membrane proteins that had hydrogens on them. And so they were able to get a picture of what's going on with a membrane protein in a living cell, able to cut through the mess of all of those atoms to just see the pieces that they cared about within a system that was living. It's a fascinating technique made possible by the resources of and that pulsed neutron source at the Spallation SNS user facility here in Tennessee. And so over the last few years, since coming to Oak Ridge in January of 2018, my role has been as the director of the Joint Institute for Biological Sciences, where I was challenged to imagine a new focus for JIBS that moved us away from biofuels and bioenergy and towards a focus on biomedical research, still capitalizing on the historical strengths of Oak Ridge. But in this case, biofuels and bioenergy have been really embedded in the work of the lab and in the University of Tennessee with whom we partner. And so really trying to go back to our roots with the Manhattan Project, further build out that biological research capability in the context of biomedical research. So I spent a wonderful couple of years meeting people across all of the directorates and organizations within the national labs, meeting people across the breadth of skill set at the University of Tennessee system, here at the flagship campus in Knoxville, but also at the UT Health Science Center in Memphis, and at a whole host of other UT-related organizations across the state, and really understanding the capabilities there and trying to build a strategic plan for how JIBS would shift to JIBS 2.0 with this new focus on biomedical research. And we won't go into this today because this is where we come on to part 2 of the talk. And then the world changed. And we've all been living that change since last year and working to figure out how we can have a positive impact and how we can have a positive shift in the outcomes that we see from this global pandemic. Department of Energy, just like all of us as individuals, was eager to understand how could we work together to make a difference for what's going on in the world as a result of this crisis. And so that in March of last year, the Department of Energy Office of Science launched the National Virtual Biotechnology Laboratory. This is a consortium of all 17 of those DOE national laboratories that you saw on that earlier map earlier in the talk, and bringing all of them together to focus on a response to COVID-19 with funding provided by the Coronavirus CARES Act. And so I've put the web link in the slides. Feel free to go dig in great detail to the vast, rich set of stories coming out of that work. I will say that as at the high level, we can tell you that there were 5 projects that were launched early in the pandemic to bring together all of these capabilities across 17 national labs to do 5 different projects. So the first is around epidemiological modeling. How can we bring together multiple data sets not just the kind of data sets that you see available at New York Times or Johns Hopkins or all of these other resources, but other data sources that exist within the DOE space, more than 20 data sources brought together to understand what's happening with the virus, to be able to create situational awareness for decision makers who need to make decisions about what's going on. The second project that I will mention here is the manufacturing project. Huge supply chain challenges with personal protecting equipment and with medical devices and with so many of the things that we needed. And so this was a group of the national labs coming together to solve problems around that manufacturing supply chain and transition those solutions into the public and into private enterprise and into companies so that they could deliver those technological developments into the real world of practice. And so just a couple of highlights there. Working with the Coca-Cola bottling company to transition, take the small presets that they create that will eventually get blown out into Coke bottles to deliver Coke to our doorsteps. And instead, repurposing a small portion of that flow so that it can be used for transporting viral media, so that you have the test tubes that you use for all of those sticks that have been going up our noses for all of these months. Or working with the carbon fiber technology laboratory to reconfigure processes for making the N95 material and transitioning that to a company that had never made that kind of material before, so that there was a whole new US-based platform for delivering the materials that are so necessary for creating the masks that are part of our personal protecting equipment. There was a project on testing R&D and understanding how to do the diagnostics right for identifying who has COVID and who does not, and a project around viral fate and transport. How does the virus move around within a school bus with its windows open, or in a confined space with windows closed? How can we understand how the viral particles are interacting in that environment? And finally, underpinning it all was a consortium across technology companies and all of the labs in the DOE leadership computing facilities to make available our high performance computing infrastructure to researchers outside of the national lab system for doing work that directly impacts COVID-19. Today, though, I'm going to focus on one of the 5 projects that kicked off in early spring of last year, the Molecular Design for Therapeutics project. The NVBL Molecular Therapeutics project was a partnership of 9 national labs. You see them listed here. I can tell you that we managed to hit every one of the time zones across the continental US, which did occasionally create scheduling challenges for us, but also brought a richness of skill set and people capabilities to the work that we're doing here in this project. And the goal of the project is to leverage the world-leading capabilities of the Department of Energy National Labs. We've talked about high-performance computing. We've talked about the neutron source, and the labs are also home to all of the X-ray and other light sources that serve the nation and the world, and the user facilities that enable us to do high-end research at chemical, biological, and analytical sciences. So really bringing all of these capabilities and resources together in order to identify experimentally validated leads for targets across the entire coronavirus life cycle. And so our goal is to identify spots where we can shut down the virus at multiple places along the pathway. The goal is to, so if we step back and think about viruses, they're not really alive. They're really machines that take advantage of the human physiology to replicate and grow and be released to reinfect other cells. And what we've learned from antiviral research over the years is that you're more likely to be able to shut down that non-living thing if you do target multiple places in the viral life cycle. And we were supported in that effort by the existence of lots of structural information through our own light sources and the work of others across the world that give us starting points for doing this work. And so our goal is to find as many experimentally validated leads as we possibly can for as many targets across this entire coronavirus life cycle. And to do that, we capitalized on capabilities that had been being built over the years in the national lab space, in high performance computing and in experimental techniques. And those all made use of a computational and experimental design platform. We start in all cases with inputs that are structures of antibodies and spike protein, or structures of, or structural models of viral proteins. We started with multiple antibody templates that were known to bind to comparable proteins in the SARS-1 virus or the MERS virus from previous years. We started with large databases with lists of small molecules that are purchasable. And all of that becomes input into this integrated platform that is anchored by physics-based and artificial intelligence-based models that assess the design fitness for an antibody or a small molecule that might be interacting with a protein, and iteratively feeding that into structure and sequence-driven generative models to iteratively design new molecules that we hope will be more promising as potential therapeutics. Feeding into those models are an understanding of the structure and the binding affinity, but also an understanding of the physical and chemical properties that are important for drugs, and models for techniques for modeling some aspects of whether these molecules will be safe and will behave properly once they're dosed into humans. And so there's this computational infrastructure that is making use of all of this information. And it is iteratively feeding into biochemical assays, chemical synthesis, and into structural biology to understand what's going on with the crystals, with how those molecules are interacting together in these protein systems. And as outputs of that system, we end up with designs of antibodies, designs of small molecules, where we have experimentally validated that they have the desired activity on the primary protein target and are likely to have the desired biological effect, and hopefully will have the good physical and safety parameters that we want to see in molecules that might become drugs. I'll give you a few examples of that kind of work. First is in the area of computer-aided design of therapeutic antibodies. So at the starting point, our colleagues, particularly at Lawrence Livermore and Sandia National Labs, were starting with structures of the spike protein from the coronavirus and antibody structures from the previous SARS outbreak, SARS-1. And so these antibodies bind and neutralize the spike protein of SARS-CoV-1, but they don't bind to SARS-CoV-2. And so the team used an artificial intelligence-based method that allowed them to model the modification of 31 specific locations on that starting antibodies structure. And that leads to more than 10 to the 40th potential combinations. And then they were using a Bayesian artificial intelligence method to select from out of that pool and to optimize within that pool the ones that are most likely to shift the selectivity from SARS-1 to SARS-2. And as an outcome of that work, they were able to identify experimentally validated designed antibodies. So they designed the antibody, a collaborator outside of the national lab space made those antibodies, and our team tested and confirmed that the antibodies bind to the SARS-CoV-2 spike protein, and some of them even go on to neutralize the entry of a pseudovirus into a human cell. So really making progress towards something that could become a therapeutic down the road. In the small molecule space, we had a comparable set of platforms capitalizing on the docking calculations that we've mentioned before, along with quantum mechanics rescoring and high-end high-performance computing and simulation. And so we had structures across a wide range of viral targets, some of which were generated within the National Lab Complex, others that were available publicly. And we were able to dock molecules against more than 100 different discrete conformations of those the active sites of the protein targets, and millions of molecules in the data set. So millions of molecules across 100 different targets. We're talking billions and billions of calculations, both the faster docking calculations and the higher compute load, more detailed calculations that allow us to refine our expectations. And then those molecules that rose to the top of the list, we actually bought molecules, brought them into our team, and tested them. We tested them in two ways: in biochemical assays, where we're just asking about enzyme inhibition, and also in cell-based assays of antiviral activity. So in this cell-based activity, what you do is you have the compound in a mini well plate, you introduce the virus, and you introduce the virus to those cells. Let's step back a second. You have a plate that has human cells in it. You add compound to that, and then you add in the coronavirus to it, and you look at what happens in each of those plates. If all of the cells disappear, like on the left, what that tells you is that your molecule probably is working just by killing cells indiscriminately, and is probably not a useful tool for you to move forward. If like in the middle circle, you still see cells growing and you see cells dying and virus growing and replicating, then you know that that molecule is not working to inhibit the viral infection. And finally, if you see the human cells growing and culturing and behaving well in this, but without that viral infection going on, then you know that the molecule is likely has some antiviral activity that you can work with. And so from this work, for the antiviral assays, one of the outcomes from our team is that from that list of molecules, we did the docking against at least 15 viral protein targets, 100 different active sites. And from that, we selected just over 1,000 molecules that we purchased and sent into these antiviral screens. And from that, just over 1,000, we identified between 50 and 60 molecules that actually do have a measurable impact on preventing the infection of the human cells by the small molecule. So we had 56 hits to work with. We've since gone on to follow up on those hits in confirmation assays, looking at the actual titration curve of how the molecules interact with the system and how they protect the human cell. That has taken us down to a small handful of lead series that we're now following up on to ask, do they actually have this antiviral activity because of the reason that we proposed from the computational results? What is the mechanism of action by which these molecules are working? And can we move them further down the path towards potentially becoming something that could treat disease? Separately, we've been taking a very special focus on the SARS-CoV-2 proteases. In contrast to something like HIV protease, which is an aspartyl protease with a negatively charged pair of aspartate residues in the active site, the SARS-CoV-2 proteases are cysteine proteases, with the sulfur atom of a cysteine being really important in the mechanism by which these proteins work. And it is absolutely true, therefore, that these proteins require special computational workflows. Many of the cysteine protease inhibitors that we know of in the world interact by actually forming a covalent interaction with the catalytic cysteine. And so we need a special computational workflow to work with these molecules and work with these proteins. And so the team has developed a computational workflow that enables us to take a database of molecules, identify electrophiles within that set that are likely to be able to interact with a sulfur, transform the 3D conformation of the small molecule to form that covalent bond, and do the docking to identify the covalent orientation of those molecules in the protein, and then flowing that into a rescoring using quantum mechanics, because anytime you start making and breaking bonds, you need to move from classical mechanics into a more quantum-based perspective on what's happening with those. And so we've been working with both the main protease of the viral protein, and also that's the one in the top here, and with the papain-like protease that also has activity as a deubiquitinating enzyme. And so we have early results out of those two activities. In the upper level, we have identified a molecule that actually inhibits the protein at a less potent level, but still inhibits the protein in a non-covalent way without making that covalent bond. And we've identified some early hits that inhibit the protein and do form that computational bond. And so they've actually used our workflow to identify molecules that do bind to the protein covalently and inhibit it. And those are moving on into antiviral assays now. For the papain-like protease, using structure-based design and these computational tools, they have identified a molecule that is a very potent inhibitor of the papain-like protease that does have antiviral activity in that antiviral assay that we've described before, and are moving on to optimize that to make it even better, in the hopes that we'll be able to run a very small animal study before the end of the project to ask, does it protect in an animal model of disease, does it protect from infection and from the particular ravages of serious disease? And so excited to be able to say that we have hits from the antibodies, from the antiviral screens going after small molecule inhibitors and for both of the two proteases in the virus. And on top of that, we had a part of the team whose role, we have a team that has lots of activities going on around computation and synthesis and experiment and structural work. And it's spread across 9 national labs across the US. And so we also had a team who's building the computational resources for us to be able to track what's happening with all of those molecules and understand what's going on with the data so that we can manage all of those processes. And so what has been a delight for me in particular is seeing the depth of the skill set in the national lab space. This was a team of more than 150 people across these 9 national labs who dropped their day jobs, leapt to the battlements, and began to battle against this virus and work together as a cohesive team to do this work. And so I can't imagine that you can read all of these names, but it was an amazing team that I was really privileged to work with across all of these national labs. And the impacts of that molecular design team were that we were able to form quickly as a team, to bring together the breadth of expertise to solve new structures of viral proteins, to build a huge number of computational models, to use the massive supercomputing resources of the Department of Energy to identify and design potential hits. We were able to develop biochemical assays and use them to validate computational predictions, to experimentally characterize active hits, and feed all that data back into improving the computational models. And we were able to obtain experimentally validated antibody and small molecule hits. We're now actually at 9 months, 10 months into the project time, but the starting hits were identified within 6 months of time. And by the time this 1-year project has completed, we will have advanced those molecules even further. So it's been an amazing team to work with who's done a lot of amazing work. And at root, the main message is that by bringing together this collection of labs, we were able to work together to do things that no single lab could do alone. This crisis has taught the world many things. And one of the most important, I think, is that it's important for us to collectively face the challenges and respond to this. And the DOE partnership has been just one model of that. And with that, I'd like to thank you for your attention. Thank you for the time. Thank you for the invitation to participate in this lecture. And I can take any questions that the audience might have.